Hello, my name is Jacob Clark, and I am a junior biology major on the pre-med track at John Carroll.
And I'm Katie Vasilauskis. I'm a sophomore biology major, also on the pre-med track here at John Carroll.
This summer, we worked alongside Dr. Aaron Johnson in studying innate immunity. Our lab works closely with how the drug PALA enhances antiviral protein production in type 1 interferon signaling, which plays a major role in the suppression of viral replication. Type 1 interferon signaling is one of the 3 different pathways within the JAK-STAT pathway, the main response mechanism for the body's innate immune system when fighting an infection. It had been previously discovered in our lab that PALA enhances the transcription of antiviral genes when coupled with interferon-beta, which is the specific cytokine that activates the type 1 signaling pathway. PALA does this by increasing the expression of the ISGF3 protein complex, which binds to a specific DNA sequence in the nucleus called an ISRE element, leading to the transcription of antiviral genes. Previous research suggests that type 1 interferon signaling is also linked to the production of the STAT1 homodimer, the protein complex formed by type 2 interferon signaling pathways. The type 2 interferon signaling involves activation by interferon gamma, which produces the STAT1 homodimer. This protein complex binds to a specific DNA sequence known as a GAS element, which leads to the transcription of different antiviral genes than the ISRE element. Since STAT1 homodimers are also produced by type 1 interferon signaling, our experiment this summer aimed to determine if PALA also enhanced GAS element-mediated transcription of antiviral genes in combination with interferon beta. I will now turn it over to Katie to talk about our hypothesis and our approach to this research question, as well as what we found.
Thanks, Jacob. So our hypothesis was that our GAS-GFP reporter cells, which were treated with interferon beta and PALA, would show greater levels of expression compared to cells treated with interferon beta alone, since the PALA would enhance transcription via STAT1 homodimers. Our approach for testing this hypothesis was to insert a virus into raw 264.7 cells, which are mouse macrophage cells. The virus infected this cell with a DNA sequence called a GAS element, which is the DNA sequence that the STAT1 homodimer binds to activate transcription. This DNA sequence was tagged with green fluorescent protein, or GFP, and it also contained a gene for puromycin resistance. After inserting the virus into the cell, puromycin was added to the cells to kill off any cells that did not take up the GAS-GFP reporter gene. The cells were maintained in puromycin for 3 weeks, and after that, they were plated on glass cover slips, fixed in methanol, and stained with an anti-GFP antibody. The cells were looked at under UV light, and as shown on our poster in Figure 4, standard background fluorescence was observed. Some cells were left untreated, but others were treated with various concentrations of interferon gamma. Additionally, there were also some cells treated with just interferon beta, just PALA, or a combination of interferon beta and PALA. A fluorometer was used to measure the GFP signal from these cells, and the results are shown on our poster in Figure 5. It's evident from these results that the cells were completely unresponsive to interferon gamma, since the concentration of interferon gamma had no effect on the strength of the GFP signal. Similarly, the cells treated with just interferon beta, PALA, or both interferon beta and PALA all showed similar levels of fluorescence. This data led us to conclude that the viral particles, which we initially infected the cell with, did not contain GFP under control of a GAS element. So future work will be aimed at generating another GAS reporter cell line, and work has already begun with a new lentivirus containing the gene for luciferase under control of a GAS element. Thank you for listening to our presentation.
Thank you.